Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our next talk, Overcoming Challenges in Manufacturing Immune-Related Transmembrane Proteins to Accelerate Cancer Research. I'm pleased to welcome Tracy Zhao as your presenter. Tracy is the West Coast Field Application Scientist at ACROBiosystems. Tracy is working with leaders in both industry and academia to support their research needs. Tracy is part of a team that helps to guide the use and development of recombinant proteins that can be used for COVID-19, CAR T-cell therapy, as well as many other immune and autoimmune conditions. A warm welcome to you, Tracy. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during this presentation. You can also benefit from some fantastic supporting literature from ACROBiosystems, which you can download for free below this live stream player. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. And now, without further ado, I will hand over to Tracy to begin her presentation.
Yeah, thanks for George for your kindly introduction and really appreciate it. Good morning, everyone. My name is Tracy. I appreciate the opportunity to be with you today. The subject of my presentation is about overcoming challenges in manufacturing immune-related transmembrane proteins to accelerate your cancer research. So let's go to the first part. It's about our company, ACROBiosystems. Acro is a leading manufacturer of key reagents. It was founded in 2010. Our headquarters is located in Beijing, and you can see offices across the globe. And for United States, our headquarters is located in Newark, Delaware. We are able to provide our products to over 7,000 customers from more than 70 countries. Our mission is to deliver the world's highest quality research products that accelerate the development of groundbreaking therapies. There are 6 technology platforms based on structural analysis and application scenario development to make sure our products could meet customers' needs. We have HEK293 expression platform used to express difficult target proteins. Homology-assisted protein structure helped us to develop multimeric protein. Nanoparticle antigen display was helpful in developing protein-conjugated particles. Enzymology, which focuses on true enzyme development and transformation. Star staining is a site-specific fluorescent labeling technology. The last one technology is to provide our customer with full-length multi-pass transmembrane proteins. And this technology is what we need to focus on today's talk. So let's go to the second part. What's transmembrane protein and what kind of challenges in developing? As we know, integral protein come in many different types, such as GPI-anchored protein, lipid-anchored protein, single-pass transmembrane protein, as well as multi-pass transmembrane protein. However, multi-pass transmembrane protein account for as much as 70%. Membrane proteins are the main bearers of biofilm function and play an important role in life activities, such as material transport, enzymatic reaction, signal transduction, structural support, cell recognition, and adhesion, etc. These functions are critical to cells and biological tissues. Once membrane protein function abnormal, it will be associated with many diseases. So multi-pass transmembrane protein with full-length complete structure plays an important role in drug development. Some customers make curious that antibodies and antigen only bind on the extracellular domain, so why we need full-length transmembrane protein? For example, you can see the CD20. It has 2 extracellular loops here, and each extracellular loop has a specific function and even interact with each other. Full-length, it will ensure the extracellular protein and the protein with its conformation, correct conformation in biological relevant, enabling extracellular loop to be completely exposed That's to improve screening of the ideal antibodies. Meanwhile, we have fluorescent products. The GFP tag outside the target transmembrane protein, and each target protein contains one GFP, so the cost is low and easy to produce. Unlike the design for soluble proteins, transmembrane proteins development is a long and complex process. There are some challenges here. It could be summarized as the following three parts. The first one, limited surface area of the cell membrane and the functional complexity. This factor restricts its expression abundance. Second, due to its highly hydrophobic of amino acid combination, it's easy to polymerize, which seriously affects its conformation and activity. At last, inherent conformational flexibility of membrane protein make it difficult to stabilize when separated from the cell membrane. So let's go to the third part. It's about what kind of the technology platform we use for manufacturing transmembrane protein in ACRO. Actually, this technology, we give it a name. It's called FLAG. Its full name is Full-Length Active Gallery. This is our technology platform developed to directly overcome the challenges in manufacturing of multi-pass transmembrane protein. We developed the three platforms to stabilize the transmembrane protein for delivery to our customers. They are detergent micelle platform, nanodisk, as well as virus-like particle. I will introduce them one by one. The first one is DDM Warren. The transmembrane protein is very hydrophobic, and once extracted from the cell membrane, it's difficult to maintain the correct conformation in common buffers. In order to solve this problem, we use amphophilic detergent. You can see it has hydrophilic head and hydrophobic tail. It will to form the micelles in the hydrophobic region of transmembrane protein. The typical workflow of to produce the DDM version of transmembrane protein could be summarized like this. Through the screening of the expression system, we commonly use HEK293 cells, but insect cells and CHO cells can also be selected. After the screening of detergent, and finally by affinity chromatography, ion exchange chromatography, or molecular sieves, resulting in a DDM version membrane protein. This method has intact epitopes, high activity, and bench-to-bench consistency, so it could be accurately quantified. However, you can see the blue one, because the detergent participate in the manufacturing process, and we know detergents are somewhat toxic to cell membrane. So this type of transmembrane protein not suitable for cell-based IC. And you can see ACRO use different IC platform with the ELISA or SPR to verify its bioactivity by binding with different anti-drug antibodies. The next one is in nanodisk version. So how to get rid of detergent? So to make our transmembrane protein could be used as a cellular IC. In order to solve this problem, we found a backbone of lipoprotein. This backbone is composed of phospholipid bilayer and the membrane scaffold protein 1D1. After that, it's mixed with DDM version of the protein in a certain proportion, and then replaced in parallel. When detergent is removed, a nanodisk version of the transmembrane protein is successfully assembled. This method with no detergent in solution, it will retain its biological activity and natural state. Improved hydrophilicity make it has a wide range of application, such as cryo-EM, MR spectroscopy of nanodisk-bound transmembrane proteins. Studies into the transmembrane protein to lipid bilayer interaction can also be performed. And nanodisk also preserve other applications, such as functional protein analysis, immunization, antibody screening, etc. ACROBiosystems has licensed the nanodisk technology from University of Illinois, so our customer could be used with confidence. You can see we use the ELISA, BRI, and FAT-IC to detect their binding activity as well as their corresponding affinity. The last one is VLP products. For VLP products, first we need to construct an overexpression cell line. The surface of this cell line is our membrane protein. Then introduce an virate backbone. The viral capsid protein will turn this transmembrane protein displayed on the cell surface into a soluble lipid bilayer particles, then secreted by budding as a membrane-coated nanoscale microparticles. We will control the uniformity of particle size by dynamic light scattering IC. For our VLP products, its size is between 8 to 300 nanometer, which is suitable for a phage display. And the VLP-related products, we also recommend our customer use it for immunization because it has high immunogenicity. We also use ELISA assay to evaluate its binding activity. So let's go to the next part. I will introduce some specific products to let you know their applications in action. The first one is Claudin 18.2. Claudin 18.2, it was one of the first challenging transmembrane protein that we encountered. We throw software and the computer to predict its structure in order to help us predict the final product. And the screening of design and culture process, we're using anti-Claudin 18.2 antibody. The results show that the significantly improved protein in a shorter amount of time, and their binding activity as well as affinity passing ELISA and SPR evaluation. In addition, this product with high specificity toward Claudin 18.2, and you can see there's no affinity towards human Claudin 18.1, so you do not need to worry about the cross-reactivity. And the next one is about Claudin 18.2 VLP. We normally throw the FAT-IC to detect protein affinity as well as to evaluate CAR expression. You can see the Claudin 18.2 CAR cells will stand with negative control as well as Claudin 18.2 VLP protein. And the GFP signal here used to evaluate the binding activity. So the results show that the Claudin 18.2 VLP protein can specifically bind to Claudin 18.2 CAR cells. And we also use SPR to detect its affinity. One side of the Claudin 18.2 VLP protein will bind to the chip, will bind to antibody on the chip, and the other side will bind to antibody at the analyte. Therefore, the same antibody is used for binding and capturing. And based on this result, you can see the VLP of Claudin 18.2, it has a good result and affinity. We also do a study for VLP products. In this study we observed that VLP immunization has a significant immune response. You can see the red color we use the VLP protein to do immunization, and the green color is the protein immunization. So the results show that the VLP protein immune response almost 3 times in comparison to a protein immunization. And the VLP products, it has high immunogenicity. Maybe could be summarized as the following reason. The first one is intracellular domain are not expressed. That's intracellular domain binding to antibodies are not produced. Second, because VLP is more morphology, it's more similar to antigen presenting cells. That's improving immunogenicity and antibody production. However, sometimes we need to use VLP control because antibodies targeting non-specific VLP surface protein will also be produced as well. So ACRO also provides VLP S-type control associated products for our customer. Maybe they can, based on their needs, to choose. The next case is about SLC7A11 and SLC3A2. It is a heterodimer VLP protein, and it is an 11-pass transmembrane protein that we were finally able to expression. As for now, you can see it has been validated for a mean particle size between 60 to 75 nanometer to be used as an immunogen or part of biopanning. Meanwhile, its specific binding of the VLP products has been variated by using human SERC7A11 antibody, so it has good functional activity. For the QC process of transmembrane protein, we normally use fat assay to determine whether a cell expresses a protein of interest and to evaluate for expression, ELISA assay to determine the binding activity, SPR assay to evaluate the bioactivity as well as affinity. Their purity is evaluated by SDS page, and for VLP products, we also control the uniformity of particle size by DRSIC. And we also do some quality control about bench-to-bench consistency as well as stability study. You can see the lot-to-lot, we do not have significant difference. And for stability study, we normally use accelerated stability study. We will incubate our products at 37 Celsius to do stability study for almost 5 to 8 days. And you can see there's no, the function is do not reduce, so our product with good stability. So let's do the conclusion for the of the transmembrane protein. And this slide is to emphasize the feature of all three types of transmembrane protein. In general, for nanodisk version, normally recommended for cell-based IC, VLP particle products recommended for immunization due to it has high immunogenicity. And the DDM version was recommended for ELISA, SPR, and BRI-IC. In all, these three kinds of technology platform should encompass the drug development and R&D life cycle for your transmembrane protein research. So this is the end of my presentation, and I'm glad if you have any questions, I'm glad to answer.
Thank you so much, Tracy, for your insightful presentation. It was fascinating to hear of the challenges in manufacturing immune-related transmembrane proteins for cancer research and how you can overcome them. As with all the other talks, we're likely going to have lots of questions from the audience. And I can see we have received some already, so I'll move straight on to the Q&A now. Your first question, Tracy, is how do you ensure that your flag proteins are full length?
I think this is a good question because actually the product expression is designed to be full length, and we will do a sequence during the plasmid construction phase. So all this QC and the design process to make our flag proteins with full length.
It is a fantastic answer. Thank you, Tracy. I'm going to move into your next question now, which is, for the nanodisk-bound proteins, is there any benefit for the intracellular domain being exposed?
Yeah, actually for the DDM version, it means a nanodisk-bound protein. Antibody could bind to the intracellular domain, not just the extracellular domain. So as a result, the antibody produced include both intracellular domain and extracellular domain targeting antibodies. Meanwhile, the assembly of nanodisk is stimulating the cellular membrane environment, which is equivalent to helping the membrane protein restore the original stable membrane localization conformation, so it's more close to the natural state.
That's excellent, thank you. And we've got time for one final question today, which is, based on the immunization strategies listed before, which method would you recommend?
Oh, actually, as I summarized before, actually we recommended our customer to choose VLP products used for immunization because the VLP-related products, its morphology is more similar to antigen-presenting cells. That's will improving immunogenicity and the antibody production.
Thank you so much for that, Tracy. Unfortunately, that is all that we have time for today, but it's been an absolute pleasure to have you present with us.
Thank you, George. Yeah, thank you for everyone. Thanks for taking your time to participate in this meeting, and see you next time.
That's excellent. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thank you once again, Tracy, and we will be back soon with the next talk.
Okay, thank you. See you next time.